So welcome to Honeybee Watch Symposia Series. For those of you in the Northern Hemisphere, happy spring equinox. Yay! And for those of you in the Southern Hemisphere, happy autumnal equinox. We are here with my colleague Rigo, who is the science director of Honeybee Watch. He's going to be moderating the Q&A. And we have five very important guests with us today, who I will introduce in a moment. And today's symposium kicks off our third year. Amazing. It's really exciting that the longevity and the general interest of the Honeybee Watch Symposia series. So thank you to you. We have something like 237 people who, well, exactly 237 people who have registered. Let's see how many can actually show up. They are from literally all over the world. So some are probably sleeping at the moment. Some of them are actually working. But it's nice to see that already we have 51 people. So thank you for joining us. Usually I start with an introduction to Honeybee Watch, since many of you are new to us. And I'll do it very briefly because we did a longer version that was recorded and can be viewed on our resources page. Rigo, if you could just drop that link into the chat. But briefly, we are an international coalition tasked with better understanding how free-living honeybees survive via natural selection with little to no human interaction. And by free living, we mean bees that have chosen where they want to live. So it's not bees that are in boxes, but they might be in tree cavities, they could be in a log hive, they could be in a chimney, on a branch, something like that. In Honeybee Watch's 5 years, our research partners and participating citizen scientists have identified more than 2,250 nest sites on 5 continents. And with your help and other participants, we keep growing those numbers. So if you could continue helping and contributing to the data, you can do that on our online database. And Rigo will drop that into the chat as well. A few years ago, we formed a task force to collaborate with the International Union for the Conservation of Nature to reassess the red list status of Apis mellifera in Europe. Prior, it was deemed data deficient. And that was at a time in 2014 when there wasn't much known about free-living honeybees. And in the interim, there have been a lot of independent research studies that have occurred throughout primarily Europe. And what we saw is that people weren't necessarily communicating. They were not sharing data or resources like protocols and best practices. And we saw an opportunity for Honeybee Watch to bring people together, start exchanging with communications and dialogues and things like this. And as a result, we have written a report that should be published soon. We're not sure. It's already been delayed, about the hopefully updated red list status of Apis mellifera in Europe. So stay tuned for that. In addition, we put together or helped put together a consortium that applied for and was recently awarded a biodiversa grant from the EU. And that's why we're here today, to talk about free bee. But before we get to that, what I would like to do is introduce a little bit about the series itself. So we launched this 3 years ago as a means to bring everybody together in the community of free-living honeybees. And so what we've done is brought scientists together to introduce them and to basically facilitate the exchange of expertise and data and best practices, to introduce you, the audience, to their work, to allow you a forum to basically interact with one another. Because as we've learned over the 5 years, you have these emotional and inspiring stories about your interactions with observations of and studies of free-living honeybees. And also for us to come together and discuss challenging issues. And this year we start today talking about free bee. And then we'll move in June to talk about nature-based traditions and techniques in apiculture in Africa. In September, we'll hear 3 partners' current research and findings. And in the end of the year, in December, we'll explore different countries' rules and regulations concerning free-living colonies. Today, we're going to talk about the Biodiversa Grant, and this symposium is titled Biodiversa Grant, the who, what, where, when, why, and how of Free Bee. And that's what we're going to do. We're going to introduce you to the research study. We'll hear about our panelists about their visions for Free Bee, and hopefully field your questions and inquiries about it. So a little context, we worked on this for about 1.5 years, assembling the consortium, strategizing on what was interesting to us, what our research questions would be, who to add to the team, and applying. And the process of putting that all together took a really long time. And of a few hundred applicants, we were ultimately selected with 33 other multinational research groups. So we're really proud to have been awarded a 1.1 million euro grant to fund this study for 3 years. So what I'd like to do is briefly meet our guests. They've been pinned along the top bar, at least for me. And if we could do that alphabetically and just take 2 minutes, introduce yourself, what institution you're part of, maybe what your connection to free-living honeybees are. And I think that that's how we'll get to know our 5 guests. And let's start with you, Grace.
Hi, everybody. Good afternoon, and thanks for coming. My name is Grace McCormack. I work in the west of Ireland in Galway. And basically, I got involved in honey. I am actually a zoologist with an interest in evolutionary biology, well, particularly the area of systematics, which is all about classifying organisms and looking at relationships between them through evolutionary time, which includes, of course, adaptation and selection. So immediately you're thinking ding, 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 because actually honeybees are a wonderful topic in order to immerse yourself in these areas. So I was surprised around the end of 2013, I was tasked by beekeepers, asked to provide scientific support. And I was surprised at the lack of evidence around honeybees, particularly in Ireland, and the assumptions. So the assumption was that wild honeybees were extinct, as along with our native Irish honeybee, which I could find no evidence for. And as soon as I guess members of the public realized that I was working on honeybees, as what happens when you work in a university, lots of questions come in. So we were getting reports about bees in chimneys and so on. So anyway, the assumption was they were all escapees from apiaries and they weren't going to survive, yada, yada. And at the same time, we were looking at a breeding program for varroa resistance in honeybees. And so for me, I kind of was connecting dots. My first grant I applied for, I was told that wild honeybees didn't exist, so they wouldn't fund me. So like many of you who, do things for free, myself and my student had to go out and try and find wild colonies, which we did, of course, all over Ireland, and we tracked them. And then happily in 2020, I finally did get some funding for studying wild honeybees in Ireland. And so that has given me some track record. So that's what got me here. And yeah, we have a lot of data now. But for me, I think doing things on your own and trying to do everything yourself is not a good way forward at all. And thanks to the citizens in Ireland, beekeepers and everything, we've gotten a lot of data and a lot of samples. But for me, the beauty of this consortium is I have all these wonderful experts to provide more insight and broaden what we can do in Ireland and have much higher impact to look at things across Europe. So I'm really looking forward to working with these colleagues. So I'm going to learn a huge amount more from working with them and expand all of the work that the Irish people here are doing, make that even more important. So that's why I'm here.
Great. Thank you, Grace. Joachim, would you like to introduce yourself?
Yeah, I can. So my name is Joachim de Miranda. I'm from Sweden. I work here at the Agricultural University of Sweden. And I've been working on honeybees now for about 25 years or so as a researcher. And I'm a beekeeper. I keep about anywhere between 4 and 6 apiaries going. That's just as a hobby. So my speciality in bee research is naturally surviving honeybee populations. We've got a population that has been surviving naturally without, let's say, too much beekeeping interference. This is within a beekeeping context, but with no Varroa treatments, and it's been surviving by one way or another for over 25 years now. My research, and that's not just me, it's a group. Barbara Locke is also a major researcher. We collaborate quite a bit. And also with Lotta, every once in a while. Hi, Lotta. Our main question is what sort of traits these colonies, bees, have developed that allow them to survive. And this includes both Varroa-dependent traits, bee-dependent traits, and because my specialty is in viruses, also virus-related traits. And that is a continuously ongoing research field, and we never run out of questions to ask. So that's where I'm at. And then, of course, the reason why I think this is an excellent consortium and an excellent way of approaching this particular thing, this research, is that with international collaborations, especially over large geographic areas, you all of a sudden have instantly a much greater geographic and biogeographic range over which you can do your research and your interpretations. You get to meet a bunch of colleagues that you've known, and sometimes colleagues that you haven't known yet. So you get to expand your network. And just in general, I think collaborative research is much more interesting, much more rewarding, and much more complex. It allows you to add complexity to things. And I think those are all the questions that is there anything I forgot?
Nope, that's a great introduction.
Okay, thanks.
Thank you. Andrzej, if you could unmute yourself and introduce yourself.
So, hi everyone, my name is Andrzej Oleksa. I'm a researcher and lecturer at Kazimierz Wielki University. I'm based in Poland, in Bydgoszcz. Why I study free living colonies? So my interest has grown over almost 20 years as I explored ecology of beetles associated with tree cavities, and I discovered free living colonies in the same field sites, sometimes in the same trees. So I started to study their ecology, genetic diversity, and interactions with managed honeybees. What excites me the most about free bee, I think it's the interdisciplinary approach of the consortium, because it combines very different aspects like ecology, genetics, conservation, beekeeping, to better understand free living colonies. And it brings together experts from different countries. I think this is very, very good opportunity for collaboration, data sharing, for developing new research methodologies. Oh, I think that's all.
Great, thank you. Alice.
Good afternoon, everybody. Thank you for taking your time to listen to us. My name is Alice Pinto, and I'm based in Portugal, in Bragança, and I work in the Polytechnic Institute of Bragança. I have been working with honeybees for over 25 years now, and I started my career with the honeybees, with the free-living bees, studying a population in South Texas, free-living colonies that was undergoing Africanization, and I looked at the genetics of this process of Africanization. I guess I can define myself as a population geneticist and with honeybees as the model system. My interests are in understanding genetic basis of adaptation, and that will be my contribution in this freebie project. And I'm also been looking at introgression of C lineage into M lineage bees, and I'm very curious to see whether the free living bees will be distinct from managed bees. I'm very keen to work with all these wonderful people in the consortium, and looking forward to answer some questions. For example, are there free living bees in Portugal? Because anytime I ask the beekeepers, they say, oh, no, there are no free living bees. There were before Varroa arrived, but not anymore. So I'm very curious to see the status in Portugal. I think that's all. Thank you.
Thank you. And Fabrice.
Hello, everybody. So I am Fabrice Requier. I'm a French research scientist working at IRD, and I am based in Paris at University Paris-Saclay. So I am an ecologist working with pollinators and bees. So I started working with honeybees 15 years ago, doing research on ecology and especially the response of honeybees to pesticide exposure, lack of flowers, and working with managed bees, so with beekeepers and beehives. And so during these years, as an ecologist, I was more and more frustrated about the response of honeybees in beehives. Are they responding based on the natural adaptation, or what is the contribution of beekeeping management? So I started to be interested in these free living colonies as an example of adaptation without beekeeper management. And so I started that, it was in 2018, to do some small research as a curious guy. And then I started now to develop research with based on programs, standardized protocols. I started that in 2020 in France with some funding from public agencies. It was the opportunity to upscale these questions, and especially to see if what we observe in France are similar in other parts of Europe, and if we can, or with this larger scale, if we can find climatic effects or larger scale effects. So that's all.
Thank you, Fabrice. And Fabrice is actually joining us from Nairobi, Kenya, where he's doing a research study there. And he's worked also in Latin America. And I think some of our other panelists have worked internationally too. So as you can tell, it's a very impressive panel of experts in their field, and especially free living honeybees. So it's truly an honor to be working with them and to be forwarding this freebie project. Grace, would you like to introduce us to freebie?
Okay, let me share my screen.
And just for people to understand, the Biodiversa call is the European Union, but not every country participated. And so you might know of other people who are also leading this this field, but because their country did not participate, they are not part of our consortium. Grace functions as our project coordinator, And it makes total sense because Ireland has really been leading the study with something like 750 nest sites that you've been researching and identifying and monitoring for something like 7 or 8 years. So this is Grace, and she will present an introduction to free bee.
Okay, thanks, Steve. Yeah, so yeah, I have the pleasure of doing this. So I'm just going to give a short presentation to give you the kind of an oversight of the project, and then really I would really welcome the panel will come in and follow up with whatever I've left out, and then we really encourage you to ask us whatever questions you like, and we'll have a good old discussion afterwards. So the idea, I mean, behind Freebie, you know, I kind of jumped on board because sometimes in the EU, it's not often you get a vehicle, a funding vehicle where you can do this kind of work. And so this Biodiversa call was about nature-based solutions and transformative change. So I thought, this is a really good opportunity for doing this because, to be honest, with the free living bees, it really is, they really do provide the key to actually improving beekeeping, I firmly believe anyway. So yeah, so free bee, of course, means free living. It's about free living honeybees, or FLCs. You'll hear us calling them that during the presentation and afterwards. And the idea behind this is looking at free living honeybee colonies in Europe, so nature-based solutions to safeguard and promote transformative change. So it is about conservation also. It's not just about the beekeeping industry. And of course, many of you on the call are really interested in that, interested in bees for the bees' sake, not only for bee farming. There's two distinctions. Okay, so really we have a team of these international scientists who you've met, but really it's about collaborating with stakeholders. So none of our research would really be feasible without citizens and without the beekeeping citizens, as well as other people who are interested in bees for their own sake and for conservation. So that's what this is about. It's a really cool project, which I hope you will agree. Now, I think one slide. So
Great. Sorry, Grace. I just wanted to interrupt. If you have questions for any of the panelists, type them into the chat because after Grace's presentation, we'll take a couple minutes to field some questions, and then we'll start with a panel discussion. And then afterwards, if there's time, we'll take more questions. Sorry to interrupt.
no, not at all. That's great. Okay, so these are, you've heard from all of us, the different universities. These are the logos of the different universities that are involved. And there's 5 major partners as you've seen and you'll see throughout. So as I said in my brief intro, like the situation in 2014 was really the understanding that wild honeybees didn't exist, that they had gone extinct, which actually is quite shocking for such an important species, right? And of course, honeybees have this dual distinction. They're regarded as both domesticated, but we know they're not domesticated, and also wild. But then if the wild bees were extinct, this would be a very sad situation for me as a zoologist. So yeah, whatever reading I was doing, I started, and the IUCN report was the International Union of the Conservation of Nature, which has a report on the I suppose the threatened status of many different species. I was really shocked, as many in the panel, and actually I think Elise was on this panel, regarding Apis mellifera as being data deficient. So really, people really didn't know if they existed or not. Surely in some countries where there's really intensive beekeeping, it's I suppose it's predicted that wild honeybee colonies might not survive, might not exist. But is that true over the rest of Europe? So in Ireland, I came across these kinds of quotes. There are no feral colonies left because of Roa. So these were kind of beekeeping attitudes. And also then there are no wild or native colonies left because of Roa and hybridization. And this is an anonymous NPWS official for people who are not from Ireland. This is the National Parks and Wildlife Service, who are the national agency responsible for our national parks and wildlife. And I did meet with the lead entomologist there when I first started, and he was very skeptical of any existence of both native bees and certainly wild bees. So this really was the state of play. And then I guess us in the panel in the background, some of us have then been doing it in the background since then, and others are interested. So in Ireland, Brown et al., we did publish a paper in 2020 where it described 3 years of following about 70 colonies that we found that we could, with resources that we could follow. And since then, we've extended this work to show that, yeah, now we have around 700, 800 colonies identified across Ireland. And we have a whole team of nearly 80 custodians now. And we have a WhatsApp group, and we have a team that are monitoring these colonies. You will see on the right-hand side here that the majority of nests are in buildings. We do have some in trees, but you'll see interesting slides in a few minutes that Ireland is different from elsewhere in Europe because we don't have these big forests that they have in Poland and France and in other places, and Sweden certainly. So things are quite different. So we are about to publish this year. Arrigo's busy writing this paper, and we're waiting for this year's winter survival to add for 3 years. So this is the kind of some of the data we're gathering. Again, I'm not going to talk about it for very long, but this is the grant that I got in 2020. Well, I got the funding, so it started late 2021, and then we developed a platform, which some of you on the call will be familiar with, where we have the custodians monitor a lot of colonies. So each line there is an individual wild colony, and then the dates we started this schematic. Arrigo builds this for us, 2023. And so where you see red, it means the colonies died out, and you'd expect some colony die out. And where you see dark green is where there's a confirmed activity. We measure activity at least 4 times a year where possible, and where it's light green, we're presumed that they're still alive until we know that they're dead. So we've got very good overwinter survival. So overwinter 23-24, over 80% of the free living colonies that we are monitoring have survived. We're backing this up with genomic data, so Arrigo's busy analysing that and developing, I suppose, interrogating the bioinformatics approach so that we can prove lineage persistence throughout those years. That it's not just that they are feral colonies dying out every year. We are very confident of many of the colonies that they are surviving. And then my student Alex is looking at, we've started some work, which I'm really excited about, extending this work with Joachim's expertise, looking at disease, various pests and pathogens in the colonies. So far, we've just looked at about 93 different colonies, managed, free living and treatment free. We see no significant difference between most of the pathogens. The only one that's significantly different is Nosema ceranae. We have high proportions of them in actually most of my colonies, treatment-free colonies. But there is no significant, even though there's slight differences you can see on this graph between them, there are no significant differences between free living and managed. You see, interesting there, we have high proportion of Latimaria in Ireland and actually no Crithidia. So there's interesting patterns here, which I really look forward to looking at as part of Freebie. And then Fabrice has done a lot of work in France. So this is a paper he has published in Conservation Letters in 2020, which I encourage you all to read. And the first part of this describes a meta-analysis of publications of forest-dwelling bees, where they did an analysis of all of the literature, and then with specific criteria, which I won't go into in detail here. Basically, the strict criteria brought it down to 106 different sites where they predict and did some ground truthing as well, looking at these at the actual cavities. And you can see at the bottom on the table here where you see management types, so managed and unmanaged. You've got this is the Korean land cover type, CLC, so coniferous, mixed or broadleaf woodlands, and you can see the estimated mean densities of the occupied cavities. And you can see here the trend is that there are higher numbers in the mixed and broadleaf forests. So the map there gives you kind of an idea of the densities, the expected densities of these occupied nests in forests in Europe. And you can see actually there's no data for Ireland, because actually there's probably only one forest in Ireland, the Killarney National Park, that meets any of Fabrice's requirements for a forest. So this is interesting, really interesting for us to try and get more data from our forest in Ireland. We've gotten rid of a lot of our trees. So this again is from Fabrice's work, where they looked at, well, how many free living colonies can you predict that there might be in Europe? And so they estimate over 80,000 free-living colonies could be present or safeguarded in European forests. So this freebie project is really good to interrogate some of this, because you'll see actually in a minute that Portugal hasn't really been included in some of this work, and we know now that there are free-living honeybee colonies in Portugal too. So here you see 2% of the currently managed honeybee population of European countries are free-living colonies. So this graph here on the right shows you the grey here are wild colonies and the black are managed colonies in terms of density, population size. And if I'm doing this, if you've questions from this and Fabrice thinks I'm doing something, not explaining it well, he'll come in and explain it better afterwards. This graph as well then shows, again, in Ireland there's no data, and in Portugal no data, so this freebie project is going to actually add data here and improve this kind of analysis. But what you see here are predicted hotspots of honeybee nests, and where you see on the right left-hand side here for each of the hotspots labeled on that graph, you can look at whether the predicted population size is based primarily on cavity density in those areas or the size of the hotspot area. So you can see actually Sweden, it looks good for you, Joachim, that there will be a lot of free-living colonies predicted in Sweden. And we actually can add data for Portugal and Ireland. And of course, there's a lot of data building for Poland through Andy's work. So yeah, so we will be able to add a lot more information here, because also actually for those on the call who are listening afterwards who aren't again in the 5 partner countries, through Honeybee Watch, we are really welcoming further collaboration with countries who couldn't participate in Biodiversa. So as I said, Andy has been doing some work in, I call Andrzej Andy because I don't pronounce his first name properly. They have been doing since 2013, so some of this work has inspired our work in Ireland, has worked on rural avenues, tree-lined avenues, and looking at occupied nests. So they have looked at 142 kilometres of tree-lined avenues surveyed, finding 45 free-living colonies. So they've surveyed a lot of different avenues, so 201 different tree avenues, looking at approximately 70 trees in each one. So then from this they can calculate the density of free-living colonies in these avenues at 0.1 nests per kilometre squared. And also then look at the preference of bees for different kinds of trees. Here the colony showed preference for thick trunks, which you might expect, but no clear preferences observed for specific tree species, whereas we sometimes think that they prefer ash and beech and oak here rather than other trees. So that is going to be really interesting as well, to combine our work and explore the patterns across Europe, both across climate and latitude, to see what the preferences of the bees are. So we have these 3 partners that have done some work on free-living colonies and now can really learn from each other. And this is a picture of Andy's one of the tree-lined avenues, and another nice picture of some of the colonies that he's found in Poland. Nice dark bees. And then of course, so Arrigo presented this graph for us. So this is data from Honeybee Watch. So there's about 878 colonies across our partner countries so far, out of nearly 1,700 colonies in Honeybee Watch. So we have a good start. As we say, we know there are some colonies where we expect colonies to be found in Sweden. We know there are some citizen scientists looking at free-living colonies in Portugal, and then we will develop our work in the other 3 partner countries. For me, we have a lot of wild colonies, but we haven't actually explored our forests. So we have, and we want to actually look in more detail. So if you look at too many colonies, you can't do them, you can't look at any one of them in real detail. So we're going to focus in on some of our colonies in much more detail. So the aims of Freebie then are to explore the existence and resilience of free-living colonies across Europe. And then to understand, really this is fundamental, to understand the survival across latitude and climate. So we have this lovely gradient, which I'll show in a few minutes, so that we can really look at patterns in a much broader context. In that, we'll be looking at different biological, behavioral, and environmental factors that contribute to that survival, and then harness all of this information to transform beekeeping. So we really want to learn what is it about these free-living colonies that are allowing them to survive in the presence of varroa and AFB and all sorts of things, and how we can use that to improve beekeeping. So we have 6 work packages, and each of the partners are responsible for a work package, and then we will all work together to bring the data from each work package together to increase the impact. So I'm leading work package 1 with Honeybee Watch. So Steve, who you've met already, is the project manager of Honeybee Watch. I'm the coordinator, and he's our communications director and our project manager. So we are looking at really, the first step is really the discovery and the distribution of free-living colonies across Europe. So it's like the recording of where free-living colonies are by citizen scientists and ourselves. So we're developing a new online platform, an international-based platform that will work across Europe for reporting and monitoring free-living colonies, engaging citizen scientists, including beekeepers, park rangers, vets, the whole lot. And then looking at the impacts of climate and landscape on the location of where we find these free-living colonies. That feeds in very nicely to work package 2, which is led by Fabrice. And here you can see the map of the latitude, the latitudinal gradient, and then the mean annual temperature on the x-axis. So you can see we have a nice spread, and through working with citizen scientists and new partners, we may be able to fill in some of these gaps that we have in our latitudinal gradient. So work package 2 is kind of focused. It does overlap with work package 1 as well, but it's really field monitoring of these free-living colonies, the population demography at a population level across this climatic gradient, looking at to assess the free-living colony densities that we find and their survival over the 3 years. And actually, some of us will be continuing. We already have some colonies identified that we're monitoring already, and they'll be brought into this project and continued in for the next 3 years. So for me, I'm delighted that our current SFI grant can continue to run. We hope to provide, we will provide standardized protocols. So Fabrice has got a lot of expertise in beelining. We've started using automated sensors here, and then some of us have varying levels of citizen science expertise. So we'll bring that together and share all of our protocols and our approaches, so we all learn from each other and apply these methods. So some of us have never done beelining in remote areas, and that's really quite exciting. And then here again, it overlaps at work package 1 because we really want to look at the impacts of climate and this latitudinal gradient on survival. So work package 1 is kind of where they are, and then work package 2 is more the density and the survival. Work package 3 then is led by Elise, and this is where we apply genomic and phenotypic approaches. So this is, we will be sequencing genomes and providing other kinds of molecular data and also physical data from the bees to look at the diversity patterns across this gradient, adaptation or selection. So the picture here on the left shows a genome scan. So you can see the 16 chromosomes on the bottom here. And you see basically this is a scan across the genome to identify where in the genome you get these individual spots that might indicate strong adaptation, or this would be selection in a particular gene that might actually help that colony to survive in the wild. So we will be comparing free-living and managed colonies to look at these kinds of genomic traits. So obviously then we will be looking then at how those genomic signatures of selection have any influence on survival. Are there any patterns, as Elise said, between free-living and managed colonies? Are there patterns in different places and different categories of free-living colonies? We also will look at integration. So to develop this work on integration, so we can use genomic data to identify what is the impact of imported honeybees in different countries and different subspecies present. So via the beekeeping industry versus native subspecies, for example, and look at the impact there. And then Andy will be looking at physical comparison, so looking to see whether there are differences in adaptation of free-living colonies to living in the wild in the actual physical makeup of the bee and the combs that they're in. So the size, for example, of the, the cells, for example. Work package 4 is led by Joachim, and he has a lot of expertise, as he said, on the pathogens and pollen. Well, both Elise and he have a lot of experience in analysis of pollen. So really this is bringing together analysis of varroa pathogen, quantitative variation between free-living colonies, the genetic diversity of pathogens, and the virulence evolution of, say, viruses across this climatic gradient, and bringing in that data from the other work packages, particularly packages 1 and 2, on the climate, the landscape, the abiotic factors, the environmental factors that are present. So here, cavity characteristics, volume, entrance, height, insulation. Are there differences in the foraging patterns of honeybees, in the pollens that they're bringing back, for example, and in the landscapes they're in, and, you know, bringing that together in a lovely big meta fancy analysis that he can do to really give us huge insights into what's happening. And then work packages 5 is really where we actually apply this knowledge for the benefit of society and beekeepers. So this is our nature-based solution. So what can we learn from free-living colonies that's of relevance to beekeeping? And in the first instance, we will be actually releasing surveys pretty soon that we hope people will fill in to assess what are the surveys, the attitudes to free-living colonies from beekeepers, because we know that they vary hugely between some beekeepers who actually quietly have been taking them as stock for their apiaries for many years, and whereas others actually, in some countries, they're instructed to kill free-living colonies because of the disease they carry. So really it's going to be so cool to actually get in there and tease out attitudes and also legislation across Europe. And then we can use the information in terms of what we learn from free-living colonies to look at whether free-living colonies are a good survivor stock for beekeeping. So we are going to do a pilot on taking colonies from established free-living colonies and see how they perform in a box, and then really use that to promote conservation of native subspecies and local bees. I guess that's the dialogue now, is really we want to, we know that local bees are best for the environment and they're the best bees to keep. Anyway, so that's going to be really good, and this is where really there'll be a lot of interaction between the scientists and beekeepers and policymakers and legislators. So this is going to be, I think, a really impactful work package. And that's led by Andy. Sorry, I didn't say that. That's led by Andy in Poland. Helped a lot as well by Steve. And then work package 6 is really about stakeholder engagement. So this will be primarily driven by Steve. I'm nominally the work package leader, but Steve is our communications director. So this is where Honeybee Watch also comes to the fore. And this is communication within the team. And already Steve is whipping us into shape on a regular basis and actually has organized this whole event. So thanks to Steve for this. So communication within the team and communication with external stakeholders is really important. This is fundamental for any kind of impact from this project. So we want to raise awareness, disseminate all our findings, and influence policymakers. So everything you might expect that a project like this should do. So I'm not going to talk about this for very long, and this is only to show, I suppose, to reinforce the fact that we are really working together on this. The work packages are not discrete work packages all just doing their own thing. All of the work packages feed into each other, so that the power in this team of people and the work that we're doing is actually in bringing all of our work together and making, what is it, added value, isn't that the term? So really integrating all of the data that we receive from the different work packages into a major kinds of analysis to look at the impacts on survival basically of free-living colonies and how we can use that information. So knowledge transfer is really important for this project, and I think I have been shocked really at some of the assumptions made and the lack of attention on such an important species. So really we want to, and actually some of the attitudes of entomologists, conservation biologists, honeybees, for me, I get the perception that in some ways they're viewed as kind of as almost pests in some environments. And really, like, particularly local native free living colonies are part of our wild biodiversity. They perform an incredibly important task. So some of what we want to do is really to improve understanding of honeybees and the role they play in our environment, and the damage that they don't do, right? For beekeepers is improving attitudes for free living colonies that, you know, really, and that's through information transfer. It has to be better understanding of what are the pros and cons of free living colonies in your area, understanding the benefits of free living colonies, and there might not be benefits in some places, right? That's the reality too. So in some places, free living colonies might well be escapes from nearby apiaries. We will see. And we want to share the knowledge of scientific methods, because I think interactions between scientists and beekeepers really improves. It's about getting science out there to everybody, and for beekeepers to be able to understand what we're talking about, and we're able to understand what beekeepers are talking about. Actually, everybody on the panel are beekeepers also, so that's a plus. And of course, citizen scientists, beekeepers are part of citizen scientists, but we have the broader public who are interested in conservation and interested in biodiversity, and they are very welcome and very important parts of this project. And we can exchange information on the importance of honeybees, the importance of biodiversity, and also the scientific approach. So nature-based solutions, I want to stress it again because it's the fundamental part of both this Biodiversa call and a very important part of what we're trying to achieve. This picture is my apiary at the University of Galway, and I have never treated, I've never used chemical treatments, and I don't feed unless I have, I don't feed any of my colonies, my major colonies. So, and this stock, this colony here, for example, is from a wild, a swarm from a tree in 2018. And these are daughters and other wild swarms from free living colonies from various places. So really, we do have Varroa in this apiary, and we have varying levels of Varroa. Sometimes we have very acute episodes of Varroa, and then we have a more chronic type of a pattern. And we do see grooming. So this picture is just from one colony, about 30%, between 30 and 50% of the Varroa on the bottom boards will be chewed. So we want to look at the disease burden of free living colonies across partner countries. What are the pattern of disease in free living colonies? Are they a threat to local managed beekeepers? Can we understand what contributes to the survival of free living colonies, and then show how they can be used for beekeeping? Because we have shown, well, they can, and now we want to get performance data, gather data and distribute data about how these bees perform when they're in an apiary situation. And then finally, really, the shared experiences, protocols, and honeybee colonies between the project and beekeepers will help improve the environment by reducing the numbers of beekeepers who overuse chemicals in their beekeeping. So we really want to, there is more and more people using treatment-free, or whatever you call it, Darwinian beekeeping, natural beekeeping. There are lots of terminology now, but we really want to explore that and provide data to support that movement. These are our funders, so big thank you to Biodiversa Plus, the EU basically co-funds this whole programs and the 34 projects that Steve mentioned, of which this is one. So the Biodiversa Plus is the European platform or program that we apply to, and then each of the countries co-funded it. So in Ireland, the EPA, and this is the Portuguese funding, the French funders, the Swedish funders, and the Polish funders. So each of the local countries part funded this. And finally, this is a QR code. If you want to get involved, this links you directly to our webpage, where you can complete a very short just a dialogue box, a contact information box, where we would love if you do join in there, tell us where you are, who you are, and if you'd like to be involved in whatever level you'd like to be involved. Just write whatever you think in that box and where you fit in. And then Honeybee Watch has been, as Steve said, integral to bringing us all together. So these are the Honeybee Watch websites. So I leave that up there for a minute. Well, actually, I could stop sharing and it'll still be visible, I guess, right? Steve, is that, do you want to? So I'm going to actually just say that I've presented it here. So I guess we're going to first open the floor to the other partners to see if anything I've said triggers anything, or they want to fill in a gap that I've left out. They're very welcome to jump in.
Thank you, Grace.
Stick to time, Steve.
Yeah, more or less. We're almost at the top of the hour. So, of course, there are so many more details and intricacies of Freebie, but we wanted to just give you a brief overview. We'll start with any of the partners who want to contribute to what Grace had just presented. And then we're going to go to your questions from the audience. If you have any, please type them in the chat. And then we'll start the panel discussion afterwards. So do any of the partners want to add to what Grace said?
No, I must have done a good job, did I?
You certainly did. Joachim, it looked like you, yeah, just unmute yourselves and comment.
Yeah, this is an excellent talk. And it really describes Freebie very well. So I'm the virologist and the pathologist, and there's been a development in the beekeeping industry, I think, because that's where most of the bees are still kept, where we've seen a replacement of different virus strains, and that more acutely and more aggressive strains of the main virus that is transmitted by Varroa in beekeeping is increasingly dominated by deformed wing virus B. It's a substrain that is much more better adapted to transmission by Varroa. And this is causing concern within the beekeeping industry. Now, we don't know quite where that is in Sweden, but it is one of those, it's an element of the questions that we ask, which is basically, are there things that we are doing in beekeeping, inadvertently doing in beekeeping, that may exacerbate some of the problems that we find from a pathogen perspective in this case. And that is one of the things that I would like to find out, would like to test in this project, to see if there is some difference between the virus composition, so to speak, in free living colonies compared to managed colonies. And if that that's something we're going to find out, if that difference can be associated with the reduced virulence, and what that means for beekeeping. So that's kind of a little bit of an extension of what Grace mentioned in the talk.
Well, it's interesting because we're finding, so I have treatment free, right? And I minimally manage. And so actually discussing with Mick this morning about the bottom boards. So we're kind of doing Varroa drop, and so I'm wondering if we can improve treatment-free beekeeping, because so Mick is saying that I find debris and the bottom board gets dirty, and he's like, well, why is that happening? In the wild colonies, in the log hives, you have a really clean floor, so the bees are cleaning everything out better. So am I actually doing something, if I'm taking bees in the wild and I'm putting them in a box, am I creating neither fish nor fowl? So I'm not treating them, and I'm not cleaning everything, but neither are the bees able to, because we've got this open mesh floor, and maybe a box isn't ideal. So are there actually, I'm going to really learn from this in terms of, can we, we're just saying maybe in a couple of years we can do better design if we're going to do treatment free. Well, this is a kind of a design of a box that you should use for treatment free, that the normal national hives we have in Ireland might not be very conducive, that you need to do better by your bees, because we're seeing high Nosema cerana, high Lotimaria, and so what am I doing to my, and I have lots of chocolate too in the west of Ireland. So yeah, I'm hoping to learn from what you're going to tell me, Joachim, from these kind of colonies. It's going to be really cool, actually, to understand better the treatment-free side of it as well. You know, if we're advocating for not treating your colonies, we have to give good advice, right, from our experience. So.
Fabrice, you unmuted yourself.
Yes, yeah, maybe to add some things about biodiversa. So as you explained, Grace, it was open for some European countries. Unfortunately, for other countries, it was not possible to join. But in Europe, we have several works that was published in Germany, and that are precious and also very innovative and originals. And so the work that was especially or several papers that was published from Patrick Kern and Benjamin. And so this was very great, and located in Germany in some forest, and also one paper in Spain. But unfortunately, yes, we have a big lake of data in other parts. And so Germany was not in the list of countries for biodiversa. And the second point that I forgot, sorry, maybe I will remember later. I forgot.
We can always return to your questions. Any of the other, Alice?
Yeah, just to add about the genetic part, I'm really curious to see whether introgression is part of these colonies. For example, in France and Poland, there are lots of C lineage colonies that are used by beekeepers. And I wonder if these genes coming from C lineage are maladaptive for free living colonies. So it will be interesting to see whether they kind of select out these genes coming from C lineage or not, or whether they might be as well adaptive. We don't know. So we will use these selection scans across the genomes to see this kind of patterns. So this will be a powerful project, especially because we also have a latitudinal gradient that we can also look at the genetic basis of adaptation to different climates, from the warmer and drier in Portugal, and the cold long winters in Sweden. But we'll see how genetic architecture, whether it plays a role in terms of let the colonies survive for longer periods. Yeah, just want to add that to Grace's talk.
Thank you.
I remember, Steve.
Good.
So
Go ahead.
don't have to forget all of the work done by Tom Seeley. That is a huge a big book published on that, and it was very also useful for to design the project and to define a bit hypothesis. So all of this work done in US, in other contexts where honeybees has other issues and pressures and status also. So yes, there is also knowledge from other continents that are useful to define hypotheses, and that was useful to design the project. Yeah, just for that.
Thank you for adding that. And yeah, of course, Tom Seeley has been studying free-living honeybees for 40 plus years in the forests of upstate New York in the US. So he has been an inspiration for many of us, and possibly one of the first to be documenting these, especially for so many years, pre and post Varroa. Alice?
Just because during my PhD, when I did the study in Texas about this free-living population, so before there were European bees, and then the Africanized bees arrived, and there was a replacement of the genome. And it was interesting to see that the C lineage alleles were wiped out. Of course, there was a background in the admixed population, but it was interesting to see that M lineage alleles, they kind of maintained in the population, so probably providing some advantage. And so it was an interesting finding that let's see what we find here in Europe.
Exactly. And hopefully this will inspire other groups to, form consortia as well, and maybe apply for funding and study their free-living populations. So, Rigo, are there any questions that are specifically related to Grace's presentation that we can start with? And what I'd like to do is maybe field 3 of those and then start the panel discussion.
So there were a few questions in the chat. Well, thanks actually for all those participating and writing questions there. A few actually asked directly about honeybee watch and possible participation. There was one question though by Nuno at the beginning, right at the beginning of the presentation. He was curious about sensors, the use of sensors in colonies for the monitoring of nests. Maybe we can start from there.
Yeah, we are including the use of sensors in this project. We've been trialing some of them in Ireland, but only recently. So they're actually a challenge to get sensors because for the diversity of the nests, and we're having a challenge at the moment with the sensors we're using in how to get them into the nest so that they are useful. But yes, we will be, it's part of work package 2, to put sensors. Each country will monitor at least as a pilot, 10 of the colonies will be chosen to have sensors to gather that data. So yeah. And actually we'll also use genomics to determine persistence as well. So the colonies will be sampled at the start and at the end of the project to show, and Elise will do fancy bioinformatics to show that the colony has survived.
Yeah, and also it's difficult to use a sensor that has been designed for a box hive. How do you insert the temperature and other kind of sensors into a entrance that is 2 centimeters wide, 15 meters high? So, we have been having this conversation a lot within Honeybee Watch as to, wouldn't it be great if there was equipment that we could use specifically for free-living bees? So it's an ongoing discussion that perhaps a couple of years down the line will lead to some collaborations and inventions of equipment. So stay tuned on that. So I've heard from Arrigo that most of the questions are related to Honeybee Watch, and we were hoping more would be specific to free bee. So if you guys wouldn't mind, I would like to start the panel discussion. So what I'd like to do is talk for about 30 minutes or so about free bee. And I'm going to start with kind of an easy question. And this is for all the panelists. So, popcorn style, if you have a response, unmute yourself and just discuss. The idea is to have a conversation amongst yourselves. So why is working within a multinational collaboration valuable to you? And I know that some of you have discussed that, but if we can kind of talk about the interdisciplinary nature of freebie and the multinational aspect of it as well. Joachim, did you want to say something?
Well, I'm trying to see how many other people are putting up their hands or unmuting their microphones.
How about all the panelists unmute themselves? And then that way it's just a conversation.
We just have a conversation.
Yeah.
So like,
Unless you have barking dogs behind you.
the big difference between between doing local work and doing international work, even at EU level, is that automatically have just by doing the same kind of research or collaborating with other people, you have this huge biogeographic area that you can include in your analysis. That includes biogeography, that includes landscape compositions, that includes latitudes, that includes seasons, that includes temperature, all that stuff. And in Sweden, you can still do that because we're a very long country and we go way up to the higher latitudes. But I think that's the big advantage for me, is that all of a sudden whatever research questions you were asking locally have this extra dimension. It also means that you tend to develop your projects a little bit more around these questions as well, whereas local projects tend to be a little more focused. And then the great thing about working in a consortium as well is you get to meet new people, and you get to meet new people with new disciplines, and so you learn. Whereas if you with local grants, it's increasing that you keep working with the same people in the same group and the same ideas in just greater detail. So those are, to me, the two biggest advantages of working in consortia. I think it's great. I wish more countries would sign up to Biodiversa. For those people who are listening, talk to whoever it represents, tell your country to become part of Biodiversa, because it's a great program. And the only reason some countries are not part of it is because their countries decided that this was not an agency that they would like to contribute their local funds to, which is a pity.
Would any of our other panelists like to chime in?
Yeah, I can. Yeah, I fully agree. The first advantage is upskilling means including new climates that we don't have in the country, new conditions, landscape, climates, but also integration, the contexts. In France, we know that we have a huge integration, but when we go in Ireland, it seems that it's more conserved. So maybe we have no chance to test that only in France. So upscaling means including a kind of other contexts, and we also so include new. It's a multidisciplinarity research at the end, because not we are all biologists, but yeah, we also share a point of view of the context, doing research, and so this is also we learn a lot. It's yeah, we learn a lot with international projects.
So I can agree with my colleagues that multinational cooperation is better than working in a single country, because we can cover wider geographical area. We can improve applicability of our findings to absolutely new conditions, and also the aspect of people with different expertise, different perspectives, and different resources is also very important.
Yeah, I mean, I just think it's we will learn more. Like it's just going to provide better data rather than people doing it on their own. Like for me in Ireland, things are quite different. So it'll be really interesting to see the dynamics of what's happening in the free living bees in Ireland compared to other countries and the different pressures, both already we can see, I'd say, climatic differences, but also the lack of forests in Ireland and how that impacts the population genetics and the population demography, and even the distinctiveness between wild and managed, because it's possible in some countries where you've got remote forests that you could have a distinct population of free living bees in those forests that are distinct from managed colonies. In Ireland, we do have localized breeding, localized populations, but there's also a lot of intermixing, and it's just the different dynamics are going to be really cool to see. And each of the panel bring expertise that's slightly different from each other. So having the benefit of their expertise applied to the Irish data and Irish samples is going to really be important for results coming out of Ireland, I think, and what we will learn. So, as well as, of course, the wonderfulness of interacting with people and going to nice places and all that stuff, but really, and learning about, I don't know, like the other side of it is the work package 5, which is the policy, the conservation, the legislators, that's going to be so cool as well, to look at how different countries, and maybe, at the end of this, we can actually bring some pressure to bear if pressure needs to be brought to bear, because we'll have the evidence to show. So I just think it's going to be fun, but actually with remarkable outputs. So I'm really excited. Yeah.
And I don't think you really went into it in any great detail, but part of work package 5, I believe, is to compile as comprehensive list as possible of European countries' laws and legislation in regards to free living honeybees. And the idea is that some countries actually protect them. Some countries actually have, legislation that says they have to be exterminated. And so, to better understand where can we put our focus so that we can educate people about, these detrimental policies that really are not grounded in science. It's fear and or, pre-existing perceptions as to, how many times have I heard that free living bees are mite bombs? Based on what? We don't really know. The science isn't out there. So if we can get the science and then based on that, see where legislation could potentially be repealed, or we can, educate policymakers, how amazing would it be to, in some of these countries, set up policies that actually protect these threatened populations. So there is an element of that. I don't know to what degree we'll actually get to that place in Free Bee. It's limited to just 3 years, and lobbying is quite an endeavor and takes a lot of resources. But at least we will have, a roadmap for understanding where could we potentially focus our efforts in the future. And actually, I was hoping that maybe this could lead into the next deeper dive into the nature-based solutions. I mean, the Biodiversa call is called nature-based solutions. And, the question is, out of Free Bee, what issues will be proposed, addressed and or solved by Free Bee? Which of those excite you? And how could they benefit you and your country? And so I throw that out at the panelists. And maybe you can just kind of not answer sequentially, but build on that kind of topic of nature-based solutions.
I just want to add another level, besides the scale, which is a very powerful addition here, is that we are looking at different subspecies. For example, in the Iberian Peninsula, in Portugal, we have Iberian bee, Apis mellifera iberiensis. In Ireland, it's Apis mellifera mellifera. And in France, you have both. But we will see whether they will find, the same genetic solutions in terms of surviving. So this is also a powerful system to look at different subspecies and to see if there is any convergence in terms of adaptation.
Nature-based solutions, anybody? Comments?
Well, it's interesting because there's some questions in the chat about, you know, whether some countries are, you know, like Rosa had asked about whether some countries lend themselves better to treatment-free beekeeping than others. This project will really answer that question really well. I think yes. And then Nuno, I think, suggested, you know, whether treatment-free, you know, if there's not resistance to Varroa, is that going to be a higher pathogen burden on other wild pollinators? And it's really interesting questions actually, and things we have to look at and things I ask myself all the time. So I think our pilot on this is, I think, really important. It's only a small pilot, but looking at the data that will come out of this will answer some of those questions, because I do obviously think my bees are they're not resistant, like so we have Varroa, they're not resistant to Varroa, so they are tolerant to Varroa, which is now the new term for resistance. I don't, so do my bees, my treatment-free bees coming from wild stock, pose a threat to wild pollinators? Actually, some would say that some of the diseases started in other wild pollinators and are present naturally as well. So I think this project will go some way to answering those questions and having those discussions by providing actual evidence. And I'm trying to, you know, generate evidence and data from what I have already, and that I'm just really happy that we can extend that via freebie. So that's one really positive thing, I think, is to answer some of those questions around whether free-living colonies are a threat to not only managed colonies, well, no, it's whether managed colonies are a threat to wild pollinators. Anyway, that whole thing that we have to unpick. But then there's also using science for the greater good, which I really like about my honeybee research, applying my science to the native Irish honeybee questions and questions about introgression and the impact of introgression and the data being used for legislation. I'm really proud of that, and I think that's what I was really concerned at a SciComm conference hearing about some countries that aren't allowed maintain free living colonies. And then I learn more from interacting. I learn more about the sheer density of managed colonies in other countries and how then free living colonies could be a threat. So I just think it's a real kernel of stuff there that that we can start applying our science to looking at the broader applied questions. So a work package 5 for me is really a really exciting work package. And I think I'm hoping 3 years is very short. So I think we will be looking at very short-term impacts, medium-term impacts, and then some of it I guess in year 2 we'll be writing the next grant application to continue, right? But exactly. I think some of these are longer term. To change policy and to change attitudes is a longer term impact, but we can certainly get started.
And I just want to add, for people who aren't familiar with what Grace is referencing, in Ireland, they have one of the last bastions of Apis mellifera mellifera populations, at least they're the majority in Ireland. And legislation was proposed in the Irish Senate a couple of years, I mean, it's a few years ago already, to ban the importation of non-native subspecies. And Grace was doing the research at the University of Galway to provide the science behind that legislation. So that's what she's referencing. Do any of the other panelists want to comment about nature-based solutions and what excites you about freebie, or what the applications and implications could be in your country?
Yeah, better beekeeping.
What does that mean?
I think it's a Grace said right at the beginning, is that, but if we study how bees do this naturally without our interference, then maybe we find out a little bit more about how they come up with their own solutions to the questions that life poses them. And if we're clever enough, we can extract that information and maybe use that to improve the way that we keep bees. And also maybe start asking the questions why people keep bees, you know, for productivity, for hobby, for whatever. And if all ways of keeping bees are equally suited for different types of beekeeping. So it's very clear that up till now, at least, that the movable frame hive, the Langstroth hive, and its imitations has been usually successful in industrializing beekeeping and commercializing beekeeping. It's very practical. Everything can be done very predictable way, but that may not necessarily be the best way for the bees. And then there may be other ways of doing beekeeping that may be not very productive, but that may be more suitable for the bees themselves. So these are the kind of things that I think might be interesting, i.e., are there different ways of keeping bees that are compatible with laws and regulations and with individual beekeepers' wishes and desires.
Anyone else? Okay, I'm going to ask questions that might relate back to the audience. Did we just lose somebody? Did we just lose Fabrice? Perhaps. So what advice could you give to anyone here who might be interested in pursuing similar research goals in their country? Because we're only in 5 countries and, you know, there's a vast world out there. And I saw already that some people were asking specifically about Africa. And I know, for instance, that they've been studying free-living bees in Germany for many years, and Switzerland, et cetera. And unfortunately, those countries were not participating in Biodiversa, but perhaps Free Bee could function as a model for others and inspire this kind of cross-collaborative and multinational collaboration. So do you have any advice for people who might want to do something like Free Bee?
Find a funder, find a funding source, which is the most difficult thing to do. I think, as Joachim has said, collaborative research is good. I think some people on the call aren't scientists, and sometimes there is, you know, there is a little bit of, you know, how the data might be used. But I think if you can kind of interact with others in a trusting, you know, I think there is a lot of trust involved in collaboration. But collaboration is a really good way of, you know, I suppose some of the research out there, getting it out there, it has to be published, or has, you know, some people are gathering this kind of information for years, but it's kind of in their own computers. And some people, I'd say in this call, and people who might listen later, will have huge data already, or good data, that by finding somebody who can help with that data could be a first step as well, finding a little team so that you're not on your own. But then it's really hard to find funding. It took me years to get any funding in Ireland, so I had to go do a lot of it myself. But yeah, it's tough. I think it's tough anyway. But I think finding somebody you get on with and that you respect or whatever, and that you can trust, and sharing data and sharing ideas towards building something, and then finding a call in one of the countries, EU or US, finding a suitable call. But God will help the US with Donald Trump now as well. But anyway,
I don't know if there's going to be funding in the US
sorry,
after
we won't go
this.
there. We won't go there at all. But yeah, that'll be tricky for a while. So yeah, But that's it, is to find somebody you trust and get on with and can share data and ideas in a safe way that and co-publish and stuff, and then find a funding source. But it can take a while, so you have to spend time doing it right. Took us a while.
Anyone else have advice? Anders?
I want to add that studying free living colonies in other countries would require adaptation to local conditions, because each geographical area has its own condition, local environmental factors, local beekeeping tradition. And I expect that honeybees, free living honeybees in Africa, for example, could occupy completely different sites than in Poland. So what we need is to adapt to local conditions, and very important is also collaboration with local researchers, beekeepers, because their knowledge could be very important at the beginning to learn how to start. And other very important thing is clear methodology, because each study requires standardized colony detection methods, standardization of data collection, and also ecological methods could be adapted to different landscapes, height structures, or bee subspecies, because I cannot exclude that different bee subspecies could have different preferences for nesting sites, for examples. That's all I wanted to say.
And actually, Honeybee Watch
Mhm.
is really trying to expand onto the African continent. And, you know, for instance, we created a monitoring protocol and realized that it's really only for temperate zones. It's not applicable to, you know, a place like Africa that doesn't have 4 seasons. And also, like you say, they've got different conditions. They're, you know, Apis mellifera scutellata, for instance, it's swarming constantly. So, you know, for them to be studying or monitoring a tree cavity might not be the best methodology to study free living colonies. So what we're trying to do is gather a lot of African bee scientists and beekeepers and others who are interested in studying their local populations and trying to figure out what would be relevant on the continent, or even, you know, in the 5 different regions. What are, you know, the research questions that they may have regionally, locally, internationally, and how do you adapt, you know, the models that Freebie and or Honeybee Watch are, you know, following? How do you adapt them to local cultural, historical, beekeeping practice conditions and kind of differences? So it's really relevant.
Steve, there's some
And
people
it is
that
also,
sorry, go on.
I just wanted to add that it is also very important to be patient, because locating and studying free living colonies can be very time consuming. So flexibility, persistence are key when you are working with free living colonies. It requires a lot of time.
May I add my 2 cents?
Of course, please.
I agree with Grace. It's difficult to get the funding, but I think there are some studies that you can do without almost funding. Of course, for genetic studies or to study the viruses, you need funding, because the analyses are very expensive. But you can do demographics, for example, without funding or minimum funding. You can do beelining to find colonies, estimate densities, and study the trees that host these colonies. So it will be a good start. And sometimes when you have preliminary data, you may convince the reviewers of the projects to fund your studies.
Actually, that's what I was going to say as well, the importance of preliminary data and getting some data in. But also, there are some questions in the chat about, you know, Are we interested in kind of what's like coordinate associated partners to this freebie? And sometimes it's very hard to get funding from the off, but sometimes when you have experienced partners and preliminary data, that can facilitate getting funding. So in terms of this group, we have to get started first, right, ourselves, and get going and discuss how, even our own interactions about publishing and sharing data. So there are things there that we have to kind of work together as a team and be on the same page about. But Honeybee Watch, and we're all part of Honeybee Watch, and so we're all up for the broader and the greater good. So sometimes if you're there on your own, you feel like you're on your own in another country, being involved with Freebie, sometimes you can get funding, because if you've got the likes of Freebie behind you, where the protocols are there and how we're doing things, that kind of support can actually also assist with getting funding. But you can start doing the cheaper stuff like I had to do for the first few years. And then there's also, sometimes it's possible to get small amounts of funding. So I got small amounts of funding from like the Eva Crane Trust and CB Dennis and the Native Irish Honeybee Society to pay for the genotyping, which is what I didn't have money for. So there are ways to get that initial data. And once you get initial data, it's easier to get funding. But also having a partner that has preliminary data and has published in the field is also really good for as a collaborator on a grant application. So that can be discussed down the line, I guess.
And also, there were a lot of countries that did not participate in Biodiversa, but because we're partnering with Honeybee Watch on Freebie, we're a global research study. So what we're hoping is that people will be inspired by what Freebie is doing and maybe participate in Honeybee Watch by, registering free-living colonies that they've encountered, perhaps following the monitoring protocol and supplying us with the data. If you're living in Europe, for instance, we would love to have help with researching these laws and regulations within your particular country. We're going to be doing beekeeper surveys kind of at the beginning of this and at the end. That's trying to ascertain what people's perceptions are of free-living colonies. So we could use help in distributing those to beekeepers, beekeeping associations, and others, veterinarians in particular. And that all leads to my last question for the panel, which is, for the people who are here, how can they get involved in Freebie? I kind of just answered my question, but
And I think we did. I mean, I think the first thing is go on to the use the QR code and go onto the website and express your interest and express what's where you're at and what you want and where you're interested in. And then I guess part of the team will engage with those people. I think that's the first step. Again, as we said, in terms of partnering and associate partners, we'd have to work that out with the team once we get started, because we haven't even had our own kickoff meeting yet as such. So, but I think the first step is to make contact with Freebie via the QR code and the website, which will I put up again, or Arrigo, maybe you could put it in the chat. And just communicate with us. And Steve there is our communications director, and he's not lazy about communicating. So so yeah, I think that's
me.
Yeah, no, exactly. I'm not even being sarcastic there.
Yeah.
So really just get in touch, and that's how we won't promise anything for a while, because we have to get going, and as we said, we've agreed ourselves that we need to actually establish our ground rules for ourselves first as a team before we can extend out and branch out that team. But it's our full intention not to keep it all for ourselves, but actually to broaden that collaboration. We might actually want to apply for a larger EU grant where we will need and want more partners. So we're not stopping here either. So we will want more partners for the future, and SMEs and whatever. So yeah, let's build a community is what I say, and through Freebie and through Honeybee Watch, and watch this space. Is that a good answer?
I like that. Do any of the panelists have anything else to add? Because if not, I think we should field a couple more questions. We have about 15 minutes for the Q&A to continue.
Been typing
Joachim?
away in the chat, sorry.
Yeah, no, that's great. There have been a lot of questions in the chat that various people are answering. So again, we encourage that kind of conversations and kind of interaction. Arrigo, are there questions in the chat that we should answer?
Yes, so well, as you just said, there are many questions that have been addressed already by the speakers here. Just one that actually pops up just a few minutes ago by Vincent, and I think will be relevant in explaining a bit more the methods that we're going to use in Freebie. He was actually interested in understanding how when you find one free living colony, how actually you can understand if there are any other colonies around, or actually how to define the density of the colony in a particular area. Maybe you guys can explain that, and also something related to it. Whenever you find a free living colony, what are you going to do? What are we going to do actually in terms of studying the pathogens affecting those colonies, in terms of viruses and other pathogens affecting them? So if you maybe can give us some more details about that.
Yeah, I can try to, for the first question, when you find one free living colony in an area, if I understand correctly, it's a question if you have one, how to measure or to estimate if there are several around. So this is yeah, we have different way to do, but in the projects we do field work, and so idea is to see if we can observe honeybees and flowers in different location in the area around the free living colony. And when we see the bee or honeybee flying or just foraging on flowers, we collect them and we do beelining. And with this technique, with the beelining at different location in the area, we can, by triangulation, we can estimate the location of the where come from these honeybees we are observing on flowers, and yeah, estimate the location of the others. Or if they are all going the same direction in a single direction, maybe the free living colony direction means that you have a single free living colony in the area. So it's what we are planning to do. So it's a lot of field work, like Andy explained before. You have to be patient. It's time consuming. It's also fascinating because we are close to nature and we understand a lot. We learn a lot doing that. But yeah, and for pathogens, I think Joachim will be better than me to answer this point.
I see that actually that Joachim was very, very fast on the keyboard and he answered quite a lot, quite extensively the question, but maybe if you want to just give an overview very quickly, Joachim, of the work that is planned for the pathogen for the rest of the audience.
We will screen, how to say, for work package 4, we'll be looking at free living colonies and their nearby managed colonies, and we'll be making comparisons between them for a range of virus traits, that is the amount of virus, and also the genetics of these viruses. And I wrote in the chat, the great thing about working with viruses from an adaptive evolutionary perspective is that viruses are pretty much the only genetic organism in this entire system that can evolve in real time, i.e., within the lifespan of a project. So it would be possible if various genetic adaptations exist, or differences exist between free living colonies and managed colonies, to be able to detect that in this project. We have, of course, monitored our long-standing population for ages and ages and ages. We have samples and data going back. We do see quantitative differences in this particular population the moment that we start adding serious beekeeping to the bees. We don't know quite why those changes are occurring. They're mostly quantitative, but not quantitative, qualitative, that is genetic. But there may be differences between free living colonies. I would suspect this may be related to the ability for free living colonies to expand and grow, limited or unlimited, as to how much virulence can develop and how much viruses can change genetically. So I'm hopeful of differences, certainly quantitative and maybe genetic.
Great. Thanks, Joachim. Actually, there is quite also, well, again, you guys are very, very fast on your keyboard and answering quite a lot of the questions already. Just a point here that I see many people participating actually from Africa. There's quite an interest to develop actually by you guys at the audience to develop more research on in Africa. Maybe just a point that would be interesting. Is anybody of you of the panelists have any research going on in Africa? I know, Fabrice, maybe you have, or maybe in general. I was thinking in terms of the future of the project. How can we actually connect what we find here in Europe with what we know or what we don't know yet in Africa regarding the situation of free living colonies? Maybe if you can add a few comments on that.
Yeah, I can start. So the situation in Africa is very different than in Europe. So in Europe, we have a much more managed colony than free living colonies. It's a current situation. In Africa, it's the reverse. So free living colonies are dominant comparing to managed colonies. And so it's very interesting. And the beekeeping activity is also very different. Still catching swarms to create the livestock. I talk about beekeepers. So the practices are a bit less intense in general. Intensive beekeeping is also occurring, but less than in Europe. So the situation in terms of beekeeping, in terms of density of free living colonies are different. So yes, I have a pilot study here in Kenya that I started last year. And I was a bit shocked. I was thinking that I will be able to do the same than in France, but of course, no, it's not possible. Protocols need to be adapted, but also our way to think. The ecology of honeybees in Kenya, I don't know in the other countries, but the ecology is very different. For a simple example is absconding. That is not present in Europe, and in Africa it's common. And absconding in all our current protocols, it doesn't matter. But in Africa, if you are not thinking absconding, you will have a very wrong interpretation of your data. So when a colony escape in Europe, we think they are dead. The colony just die. Here, no, it's not the case. They just move, and so this is another way to think. So we have to learn also to adapt the protocols and the situation also with the resistance to biotic pressures are different. So the treatments against varroa are not common, not present, but varroa is present. Genetics are totally different. We are talking about other subspecies. So yeah. So this pilot study is very interesting, but the protocol that is now under tests is very different than what we are planning for Biodiversa for this 3B project.
Before you sign off, Fabrice, Nina's asking if, in your experience, you are observing that chemicals are not being used in Kenya. She's saying that they're not really on the market.
Yeah, it's right. So in the 3 different locations, so these 3 different areas in Kenya where I am these pilot studies, it's right that the chemicals are not used and not available. So they're not used because beekeepers cannot buy them. So it's right. But it's only 3 locations. So I assume that in maybe some parts of Africa and Kenya, where intensive beekeeping is practiced, maybe they have access to these chemicals. I don't know.
So what would you say, Steve? We have a few minutes left. I see most of the last question have been addressed. Maybe if you want to.
I saw that John had a question, John Locke, and Grace was going to answer it, but John, do you want to unmute yourself and ask your question if you're here? John Locke. I'm not seeing anybody appearing, so.
Yeah, his question was related to actually hornets. And so maybe if he's there, yeah.
I saw also that, Michael, you had a couple questions. Were they answered already?
There's a hand up as well, Steve.
Yeah, I just
thank you, Grace. Yeah, thank you so much. Awesome project.
So they were answered. Tibibu, you're in Ethiopia. You have a question. Would you like to unmute and ask your question?
Thank you very much. I'm not sure whether you can hear me. Can you please confirm, because my internet is not good?
Yes, we can hear you.
All right, thank you. Sorry, I joined late because in the place where I was there was not a good internet connection, so I joined very late. My question is, because I heard many of the things that you have been discussing about, but I joined late. My question is about free living bees. In fact, some of them were answered in the chat. I asked, because for me, I'm a beekeeper in the forest. And as you can see in my profile picture, that's how I don't know whether the word keep is the right one. We keep bees. Well, we work with the bees, I think. I don't know the English term when I translate it. So for me, until very recently, that I had a chance to visit the Western way of beekeeping in Europe. Every time I read the word free beekeeping, free living bees, and I was always asking, is there another way? And that and also organic thing has been always confusing for us. So my question is, because there are a lot of things that we can learn, and I was very strongly suggested to join this Zoom. And my question is for everybody or anybody who is interested, because I'm trying to do my own little research. As you can see in my profile picture, I try different types of hives and how the bees prefer it, and then how they can protect themselves from predators. So is there any collaboration that anybody can do remotely, because you're talking about funding issues as well? Anybody who is experienced and interested in sort of mentoring me or help me or my community, the traditional beekeepers, where I can be able to send at least the data and you can tell me what to do and how. And then maybe, because I do have a lot of questions in this regard and I want to study. So do you think it's possible? Thank you.
Actually, the next symposium on June 19th is going to feature 3 different beekeepers in Africa. We have somebody from Kenya. We have somebody who works on the entire continent, the Bees for Development group that have been there for about 30 years. And we have to confirm the last one, probably from Ghana. So they would probably be able to address more of these questions from experience. Additionally, Honeybee Watch has been trying to expand onto the African continent for a couple of years. We've been in conversation with lots of different bee scientists, beekeepers, beekeeping organizations. And what we've determined is that probably at the end of May, we're going to create an online working webinar to introduce Honeybee Watch, a couple of projects, one in Ghana, another in Kenya, possibly elsewhere. And the idea is for the people that we've been in contact with to come together and talk about what would you like to do? What would you like to research? What's important to you in Africa, in the region that you're based, so that perhaps we can, advance with some meaningful and kind of, indigenous research studies. So I would certainly encourage you to get in touch. I know you have my email. So you're also on our list. So we will be in touch soon. But I think that that's really the place to participate more actively with Honeybee Watch, and probably won't be specific to Free Bee because we're just in those 5 European countries, but there will be opportunities for us to collaborate potentially.
Thank you very much.
Yeah, thank you. John, I saw that you were unmuting yourself and showing your video. Is it a very quick question? Because we only have 5 minutes left and I have a couple of other things to talk about. John Locke, that is. Okay. I'm going to do my kind of, oh, John.
No, could you hear me?
Yes.
Yeah, the question was really, is anyone in the European consortium going to be looking at the impact of Asian hornets on the free-living colonies?
We actually discussed it at some point, and we wanted to keep the Free Bee project as kind of concise as possible about free-living bees. Obviously, Vespa velutina is a predator and attacking those free-living colonies, but in the end, Joachim, I don't remember, why was it eliminated?
I think we mentioned it, but we said we would just incidental, we would observe them when we're as part of our monitoring. Is that kind of what we said?
It was included in the background description as one of the potential threats, but in the writing of the proposal and the budgets and everything, this is what we could come up with. So it's not specifically included. But as Grace said, we're going to incidentally keep an eye out for them. We don't have them in Sweden yet, but I know that England has them. Or
we don't have them in Ireland yet either. No,
but you will soon. So
we won't. Wear that.
And I know that they're in France, and I know that they're in Portugal.
Yeah,
so.
Exactly.
Actually, I'm really, it's a big problem here in Portugal, and it will be in one of our forest study sites, not in the south, but in the north. So yeah, we will, when we do the monitoring, we will collect the data for the Vespa velutina.
Yeah. So unfortunately, we are almost out of time. So I just want to thank our panelists. We appreciate you presenting Free Bee with us. Thank you to the whole audience. It's incredible that 50 people are still here almost 2 hours later. I would like to plug our upcoming events. The next symposium is about Africa, as I mentioned. It's on June 19th. Following that, we have 3 of our partners who are going to be sharing their current research and findings. They're 3 PhD students, one at Grace's University at University Galway. We have Kaylin, who's been studying bees in South Africa. She's at the University of Florida with Jamie Ellis. And we have Matusz, who was here at some point, maybe he's still here, who's going to be talking about his PhD studies in Slovakia. And then lastly, actually, the events are at honeybeewatch.com/events. Rodrigo, if you could just fix that link. And then the last one in December, we're going to be talking about policy issues. So we have Loretta Neary from the Native Irish Honeybee Society talking about her work trying to ban non-native subspecies. And we'll have others who are talking about both beneficial and detrimental policies in their countries. Again, if you are in Africa and you would like to participate in this working webinar, please get in touch with us. And yeah, we have recorded this session. It will be uploaded to our website. You can also find all of the past speakers' presentations during the symposium from the first 2 years there. You can join our Instagram, our Facebook, our YouTube channels, and that kind of wraps it up with 8 seconds left. So thank you, panelists. Thank you, audience. And hope to see you in 3 months.